Hi, everyone. The video you're about to watch is part of the Age Presents lecture series. The series is produced by the American Aging Association to provide an introduction to foundational and advanced topics in geroscience. As you watch this video, I would like for you to keep 3 points in mind that will help you understand the importance of biological aging. 1, aging is the primary risk factor for disease, including cancer, metabolic syndrome, and most neurodegenerative diseases. 2, the scientific approaches that are being used here today, such as the model systems that help researchers learn more about the mechanisms within these age-related diseases. And 3, mostly centers around you. What is your interest in aging research? Whether it's biological, translational, or clinical, how would you like to contribute to the field? And if you're not already a member of the American Aging Association, I would encourage you to visit our website to learn more about us and consider joining today. AGE members receive free access to the journal Geroscience, reduced registration for our annual meeting, opportunities to interact with many of the leading researchers in the field, and access to all of the lecture slides and the notes that are being used here today. And there's more. We have a very active trainee chapter with many opportunities for networking and career development. And last but not least, I would like to acknowledge and say thank you to our sponsors for the Age Present lectures. The University of Washington Healthy Aging and Longevity Research Institute, the Jackson Laboratory Aging Center, and the Nathan Schott Centers of Excellence at both of these institutions. Thanks for listening and enjoy the lecture.
Hello, everyone. I'm Leena Bharath. I'm an assistant professor in the Department of Public Health and Nutrition at Merrimack College, Massachusetts. Today's lecture, I'm going to talk to you about nutrient signaling in the context of aging. By the end of this lecture, you will understand how nutrient signaling influences our lifespan and health span, the key nutrient signaling pathways that are operational within the cells, and you'll see as the lecture progresses how these nutrient signaling pathways converge and lead to a process known as autophagy, which is a cellular homeostatic mechanism. And since autophagy plays a critical role in aging, as well as it's my area of expertise, I thought I will introduce you to autophagy, at least briefly. Now, we all know what we eat determines how healthy we are, our life expectancy, as well as our healthy life expectancy. So life expectancy, here's the data from CDC, 2017-2018 data for both male and female at birth and at 65 years of age. The life expectancy is described as or defined as the number of years a person is expected to live. And it's about 79 years for Americans. The healthy life expectancy is defined as the number of years that's spent in good health, or to be more precise, the number of years where there's absence of any serious or life-threatening illness. Now, healthy life expectancy is only about 63 years for Americans. Now, around that age, most older people get diagnosed with metabolic illnesses such as diabetes, metabolic syndrome, insulin resistance, or neurodegenerative diseases such as Alzheimer's and Parkinson's. And they're also diagnosed with several forms of cancers that occur with age. Now, this is a review that talks about health span, and the bar here represents the pathological age. Now, the disease-free life is shown in green color, but as aging progresses, there's increased incidences of aging-associated chronic diseases, and the bar color changes to red. Now, you can see here aging-associated diseases occur concurrently, and there's many of them occurring around about the same time or a little separated by a few years. And if we prevent the onset of one disease or eliminate one disease, the people are not free from others. They have comorbidities. But on the other hand, if we delay the onset of these aging-associated diseases, delay their occurrence, we are improving the health span. So that leads to the question, how can we delay the occurrences of chronic diseases? And that leads to another question, which is, what triggers or what signals or promotes the onset of aging-associated diseases? Now, our cells are constantly exposed to many signals from the environment. Most dominant and a potent signal that we are exposed to right from the womb until old age and death are the nutrients. Nutrient signaling is constant and we are repeatedly exposed to it. Now, cells have a way to recognize and respond to the fuel substrate, example, glucose here. Now, they are able to detect these signals and then have an intricate pathway or network that brings about a response. The pathway is within the cells and they relay these signals, and that has a functional consequence. You'll also notice these pathways are conserved among different organisms. Now here's it's a crowded slide, but the takeaway point from this slide is, if you notice, we have yeast all the way to mammals. If you observe the pathways, the signaling network that occurs in all these organisms are pretty conserved. Not only the pathways, the receptors they use to identify these nutrient signals are also conserved. Research over the years has shown us that if we inhibit these nutrient signaling pathways or alter these nutrient signaling pathways, it has anti-aging potential and has the capacity to improve health span. Now, before we move on into the signaling pathways as such, I would like to refresh your memory regarding digestion, how the nutrient signals are generated in the first place. Now, when you think of it, we consume food, and the food has macronutrients and micronutrients. Now, I'm talking about digestion of macronutrients, which begins in the mouth by the mechanical action of chewing, as well as enzymatic digestion, such as saliva has salivary amylases. So that results in the breakdown of the carbohydrates in the mouth. The food then enters into the stomach. Now, stomach, again, we have mechanical action of the stomach, the churning action, the low pH of the stomach, along with the enzymes that are present, acid-tolerant enzymes that are present in the stomach, such as pepsin, which digests proteins, gastric lipases, which can digest lipids, break down the food, and then it results in a partially digested mixture called as the chyme. Now, the chyme can then enter into the small intestine, where we have the pancreatic enzymes, such as trypsin, again, causing protein digestion. We have various carbohydrates and lipases, which can break down lipids. And we also have the brush border enzymes, such as maltase and sucrase. Eventually, the food that we ate is broken down to tiny constituents. Now, basically, carbohydrates are broken down to glucose unit, fructose and galactose, proteins to amino acids, and fatty acids, fats to fatty acids and glycerol. The intestinal lumen has the villi cells. Now, villi cells serve an important purpose in that they help in the absorption of these nutrients, which can then enter into the bloodstream. The villi cells have vessels that innervate through which the food that's absorbed can get into the blood circulation. Now, through the blood and also through the lymphatic vessels, the digested nutrients are taken to the liver and then they are distributed to different cells. Now we see protein broken down into amino acids, carbohydrates to monosaccharides, and lipids broken down to free fatty acids and glycerol. Amino acids and carbohydrates can get through to the blood circulation by blood capillaries, and the fatty acid monoglycerides would enter through the lymphatic system, and they can get distributed to different cells. Now, within the cell, you can see that glucose undergoes glycolysis, and glucose is converted to pyruvate. Then pyruvate can take either anaerobic pathway where pyruvate converted to lactate in certain cell types in certain situations. And pyruvate can also enter through aerobic respiration pathway where it goes through the, becomes converted to acetyl-CoA, goes through the citric acid cycle, the NADH generated goes through the electron transport chain, eventually resulting in the formation of ATP molecules. Amino acids as well as fatty acids can also enter these pathways at different points and then result in the formation of energy-rich ATP molecules. And I have a biochemistry book and a nutrition book present in the references, which you can take or refer and learn more about this pathway. Since this is not the focus of this lecture, I just went over just to refresh your memory. Now, what happens to these nutrients? So we have all these tiny molecules that are generated. These can be sensed by the cells. Now, once they are sensed by the cells, they relay signals within the cell, which will have a consequence. Now, any alteration in these nutrient signaling, which by the way occurs as aging progresses, can result in the onset of either metabolic, neoplastic, or neurodegenerative diseases. Now, what are the players? So the key players in this nutrient sensing network are ATP, which is generated by breaking down food, carbohydrate, smaller forms, the units of carbohydrates, glucose, for example. We'll have amino acids, which are generated from breaking down the proteins, fatty acids generated from breaking down of lipids. Apart from these macronutrients and ATP, micronutrients can signal, like zinc, for example, can signal. And we also have phytonutrients like polyphenols, which can also play an important role in nutrient signaling and in the context of aging. Now, I'm not going to focus on micronutrients and phytonutrients. We will focus on the macronutrient signaling and the key signaling pathways through which they can relay these signals. Now, three pathways are extensively studied in the context of aging. We have the insulin and insulin-like growth factor, IGF pathway, the adenosine monophosphate kinase pathway, and the mechanistic target of rapamycin, mTOR pathway. So we will begin with the insulin-like growth factor pathway. And once again, before we go into the pathway, just as a refresher, we have carbohydrates, for example, broken down to monosaccharides, which then enter the bloodstream through blood capillary, distributed to the cell. If you take a look at this image, we see that carbohydrates are digested in the intestine, absorbed through the small intestine into the bloodstream. That increases the concentration or levels of glucose in the bloodstream. So bloodstream is this central area right here. Increased concentration of glucose causes the pancreas to secrete a hormone known as insulin. Now, insulin secretion promotes a few things. First, insulin binds to the receptor on the cell surfaces. So that's an insulin receptor, which causes a signal transduction to happen within the cells. So these signal transduction will enable glucose transporters located on the cell surface to pull in the glucose into the cell for utilization, for production of ATP. Apart from that, insulin can stimulate the synthesis of glycogen, which is the storage form of glucose. Now, glycogen will be stored in the liver and in the muscles. So all these actions, but for example, the glucose uptake, as well as the storage, generation of glycogen, will cause the bloodstream to lose glucose. So the concentration of glucose in the bloodstream decreases. Now that stimulates the pancreas to secrete another hormone known as glucagon. Now, glucagon, the purpose of glucagon is to promote the breakdown of glycogen and help the release of glycogen into the blood, which is what happens here. Now, that raises the concentration of blood glucose. So this process happens in a concerted manner so that the blood glucose level can be maintained within the range of 70 to 110 milligrams per deciliter. This process happens repeatedly. And for this process, we need insulin and insulin signaling. So once again, to reiterate, so we have glucose from the blood, which is taken into the cells by the production of the hormone insulin. Now, targeted cells take by the binding of insulin to the insulin receptor, send signaling within the cells, signal transduction pathway, which promotes the uptake of glucose through the glucose transporter. Insulin receptor activation can also signal gene expression. So in detail, so this is insulin signaling pathway. So insulin receptor has an alpha subunits and then beta subunit, which is intracellular. Now the binding of insulin or insulin-like growth factor occurs in the alpha subunit. Now when the binding happens right here in the alpha subunit, there's an autophosphorylation event, meaning a phosphate group will be added to the beta subunit, intracellular beta subunit of the insulin receptor. And we can have insulin and IGF hybrid receptors or IGF only receptors. They can be autophosphorylated by the binding action of this insulin. After insulin binding and autophosphorylation of the receptors, pathways can start happening. So the pathway here is the IRS and GRB2 pathway. Insulin receptor substrate pathway leads to the activation of phosphoinosite 3 kinase pathway, which activates then protein kinase B, and here's a whole signaling run. Now, activation of this PI3K AKT pathway is pro-survival pathway, which can lead to activation of another important regulator of signaling known as mTOR, the mechanistic target of rapamycin. And you can see right here how these pathways are interconnected or interlinked. Now, activation of mTOR is an anabolic pathway, or it promotes anabolic mechanisms. So that would lead to protein synthesis. The activation of mTOR is anabolic in the sense it activates protein synthesis mechanism, but it's inhibitory to catabolic processes such as autophagy. Now, autophagy is a conserved process, and it's catabolic, meaning cell collects all the organelles that are unwanted or in excess, damaged organelles, protein aggregates, which are no longer useful for anything, and it sequesters them in a region, like in an autophagosome, a bag-like structure. The autophagosome then fuses with the lysosome, where these components are delivered to the lysosome, and acid enzymes in the lysosomes would degrade those and recycle. So autophagy is a recycling process, and we'll see a bit more in detail later on. So this autophagy process is inhibited with the insulin signaling pathway. Now, apart from this survival protein synthesis pathway, IRS activation can also result in activation of a protein called as RAC, which helps in cell migration. And we also have the other pathway, which is called as mitogen activated protein kinase pathway, right here. So this pathway results in phosphorylation of a protein called as ERK. Now, ERK kinase can translocate into the nucleus and can cause gene transcription. Now, how are these proteins activated and inactivated? So there are several ways proteins can be activated or inactivated. One prominent mechanism is called as the phosphorylation mechanism. So these proteins themselves act as enzymes, and they are capable of taking a phosphate group from ATP and add those phosphate group to other proteins and activate them. So proteins that take a phosphate group and add it to another protein, which can result in either activation or inactivation of a protein. So that protein, which is capable of adding a phosphate, is known as a kinase. And the protein that is capable, or an enzyme that is capable of taking a phosphate group, removing a phosphate group from another protein, which is capable of activating or inactivating the other protein, are known as the phosphatases. So kinases add a phosphate group, phosphatases remove a phosphate group. So with several of these reactions, activation of proteins, for example, PI3 kinase activating AKT, also known as protein kinase B, is through phosphorylation reactions. So PI3 kinase is able to add a phosphate group to an activation site in AKT and activate the protein, which will result in cell proliferation and survival and so on. AKT activation activates mTOR by phosphorylating mTOR, which results in protein synthesis. So as you can see how insulin binding to the receptor, to its receptor, triggers a cascade of events. And many of those events are important for cell survival. Now, what we, based on research, what we understand so far is when the insulin and insulin-like growth factor receptor pathway is inhibited or it's altered by either pharmacological, genetic, or lifestyle interventions, what happens is the profile changes. So when these pathways, the nutrient signaling pathways, are inhibited in older mouse model or rat model, we'll be able to see the phenotype of the mouse and it changes and start resembling a younger phenotype. Now, calorie restriction, a lifestyle intervention, and certain pharmacological agents can also mimic calorie restriction, example, metformin, have the capacity to inhibit the insulin signaling pathway. And this has been shown to be robust and reproducible intervention for extending health span and delaying the onset of chronic diseases. So as we saw in the very first, the pathological time scale, we are able to delay the onset of these age-related chronic diseases, thereby improve the health span. So some data on that, what happens is when insulin signaling is inhibited, either through pharmacological, genetic, or lifestyle interventions, we would limit the uptake of glucose into the cell. because we are inhibiting insulin signaling, which is needed for activation of glucose receptors. When glucose is inhibited, the cell lacks substrate that it can use to synthesize ATP. Now, as ATP levels drop, which we can achieve by dietary restriction as well, there's an activation of this kinase known as adenosine monophosphate activated kinase, AMPK. AMPK gets activated when there's an energy stress. When the levels of AMPK to ATP increases, there's more AMPK. AMPK binds to this kinase and activates it. So this activation is inhibitory to mTOR. Now, the mTOR inhibition had been linked to many physiological processes, such as when mTOR is inhibited, cells have improved resistance to stress. We all know, as you'll see later soon, that inhibition of mTOR activates autophagy. There's also connection with inflammation, activation of mTOR and enhancement of autophagy seem to reduce oxidative stress within the cells and reduce inflammation. So all these have the capacity to extend longevity. So here, when we inhibit mTOR pathway, we activate autophagy. Now, studies in mouse model where insulin receptor is mutated. So these are mouse that are heterozygous for insulin receptor, IGF receptor. These exhibit an increase in lifespan. Even in brain-specific heterozygous mouse model for IGF receptor, there's an improvement in lifespan in these mouse models. The mutant mouse actually lived 9% longer than the wild-type mouse. Now, there's something important regarding insulin and IGF. We all know that insulin signaling, IGF signaling is needed because you can see that PI3 AKT pathway is survival, and we see cell migration, gene expression happens by the ERK-MAP and AKT pathway. So all these are needed. So when an animal model or even humans, when we develop, we need these signals for cell survival, right? For neurogenesis, for myelination. So for various growth purposes, for growth processes, we need IGF and insulin. They also have a downside, which is when these pathways are active, now they are inhibitory to autophagy. Now, lack of autophagy or lower autophagy is linked to less stress resistance and buildup of reactive oxygen species. So there has to be a balance. So IGF has this capacity to be Dr. Jekyll or Mr. Hyde, depending on the type of signal it's sending and the time when it's sending. So some research is available that shows when IGF receptor was knocked out later in life, about 15 months in mouse models, this actually reduced the risk of cancer, but there was no real benefit in extending the lifespan. So there's a lot of research going on to identify which is the best time during lifetime for interventions, such interventions. When should we suppress IGF signaling to obtain the maximum benefit, to enhance health span or lifespan and prevent the onsets of some of the aging associated diseases. So all these pathways have this component to it. So we need those pathways, but at certain point in life, maybe later on in life, when these pathways are hyperactivated for some reason, could result in a dysregulation in the signaling network that can promote chronic diseases. This especially happens during aging. So the next pathway, and you will see that all these pathways are interlinked. The next pathway is the AMPK pathway. Now here's AMPK. Now AMPK protein is activated by the binding of adenosine monophosphate. So when the cell is under energy stress, so there's not adequate glucose intake, calorie restriction, cell undergoes energy stress. So lack or reducing ATP, but higher levels of AMP would activate this protein because AMP binds to AMPK and activates it. Apart from binding of AMPK and getting activated, AMPK can also be activated by upstream kinases. Remember, kinases add a phosphate group and activate another protein. Upstream kinases, LKB and CAM kinase. So CAM kinase is activated by the presence of intracellular calcium. So calcium activated kinase, calcium chromogylin binding activates this kinase, which then adds a phosphate group at this site, threonine 172 on AMPK, and that activates AMPK. Now, there are several research that's underway that looks at activation of AMPK because AMPK activity declines with age, and several research articles have shown that AMPK declining with age. And if we have dietary, example, calorie restriction, exercise activates AMPK, genetic ways to activate AMPK, and pharmacological agents such as metformin can activate AMPK. This is shown to improve longevity. Now, AMPK process is a catabolic process, or it promotes catabolic pathways because, as I mentioned, AMPK gets activated when there's an energetic stress, meaning there's low levels of ATP. So AMPK will turn off pathways that will use ATP and will turn on pathways that will produce ATP. AMPK, as I mentioned earlier, would be activated by the ratio of AMP to ATP or ADP to ATP increases. AMP is a central energy gauge, so it can sense energy imbalance, like a whole body energy balance control. That's how AMPK acts. Now, aging is associated with decline in AMPK activity and signaling. So if there are ways to promote AMPK signaling, then we could potentially improve aging-associated or prevent the onset of aging-associated chronic diseases. Now, apart from turning on all the catabolic processes, AMPK is also important in preventing fatty acid synthesis because AMPK is inhibitory to a protein called, or enzyme called as ACC, acetyl-CoA carboxylase. So acetyl-CoA helps in the conversion of acetyl-CoA to malonyl-CoA, which then promotes fatty acid synthesis. Now, if AMPK is inhibitory to ACC, then fatty acid synthesis is curbed. And that's another important function of AMPK because the absence of fatty acids or reduced fatty acid synthesis is beneficial because aging is associated with, excess fat accumulation, which then signals and there's insulin resistant onset. So all those can be prevented as well by activation of AMPK. Now here, once again, an overview that just shows that AMPK suppresses pathways that consume energy and upregulates pathways that will break down and utilize or promote ATP production. Now, there's research on AMPK activation. So in this manuscript, the authors were able to show that mouse that are older had severe outcomes after induced lung injury, hemorrhage-induced lung injury. The outcome was more severe. The mouse had a harder time recovering than young mouse. What they found was this inability of older mouse to recover was due to impaired activation of AMPK. The mouse actually had harder time even responding to AMPK agonist, ACAR, the AMPK activation was prevented, and the mouse had severe outcome when compared to young mouse. That shows how important AMPK in aging. So when the activity of AMPK dwindles or goes down, and then aging-associated conditions develop, that's different from a young mouse, which is able to recover from the lung injury. And another research was looked at calorie restriction on AMPK activation. So the mouse were fed, or rats were fed a calorie restricted diet, so that they were consuming only 60% of what they were consuming earlier. And this went on for 8 weeks. So that resulted in activation of AMPK. And they also observed that activation of autophagy occurred. You know, activation of AMPK can result in inhibition of mTOR pathway. So that resulted in activation of autophagy. And they found that the older mouse, when autophagy was activated, they have improved consequences. This is their model from their paper. Then they attributed this AMPK activation to improvements in autophagy, which prevented oxidative stress buildup. So as the rats grew older, they had more oxidative damage that resulted in damaged kidneys. And when AMPK was activated by calorie restriction, autophagy was enhanced, which was able to get rid of some of this oxidative damage, and that promoted health in older mouse model. So we've seen so far how insulin signaling pathway can act through various signaling mechanisms. Same way, AMPK activation can also help in preventing chronic illnesses. Now, the third pathway that we are interested in is the mTOR, mechanistic target of rapamycin pathway. And once again, there's a link between all these pathways. mTOR, we talked about it earlier, AMPK, we talked about it earlier, they're all interlinked. Now, mTOR is another master regulator of cell signaling. Now, it combines nutrient sensing signals and stress response signals, and then brings about cellular response to it. Alterations in mTOR signaling have been attributed to onset of several aging-associated chronic diseases, and you can see many of them are listed here. Now here we see calorie restriction activates AMPK, which then inhibits mTOR. Together, they are able to activate autophagy. Now, the calorie is restricted, mTOR is inhibited, mTOR inhibition promotes autophagy. This type of response was seen across organisms. This shows again that the process is conserved. Attenuation of mTOR signaling, or preventing mTOR, blocking mTOR, which results in activation of these pathways that lead to activation of autophagy, has led to, or has shown that this can lead to decrease in chronic diseases and age-related pathologies. Now, mTOR is a multi-protein complex. You can see that. There are two complexes of mTOR, mTOR1 and mTOR2. They are, you can differentiate them by the participating proteins. So mTOR1 has this Raptor and Pras40 protein. mTOR2 has Rictor and Proctor proteins that associate with this complex. mTOR2 is activated by AKT signaling and PKC signaling, and it is involved in cytoskeletal remodeling. mTOR1 is a primary nutrient sensor. We know this activation of mTOR promotes anabolic processes. So activation of mTOR can result in protein synthesis, nucleotide synthesis, activation of mitochondrial lipid metabolism. So activation of mTOR is anabolic. Now, if we see the other side of mTOR, when mTOR is inhibited, we activate catabolic processes such as autophagy. So here again, we see mTOR, and it's a dominant nutrient sensor, and it can sense amino acid signaling, glucose signaling, and it can also get the hormonal cues. mTOR2, mTOR complex 2 is activated by PI3 kinase, AKT pathways. Now mTOR1 is the activity of mTOR1 is inhibited by AMPK. We saw that earlier. And also pharmacological agents such as rapamycin, dietary interventions such as calorie restriction can all inhibit mTOR. Now, mTOR inhibition promotes longevity. So this is what research has shown many, many times. mTOR inhibition promotes longevity by activation of processes such as autophagy. Now, how does mTOR get activated? So amino acids, insulin can all activate mTOR. So nutrient signaling can activate mTOR. So let's start at a situation where there are no amino acids, no insulin, and mTOR is inactive. So mTOR is in the cytoplasm. Another important protein that regulates the activity of mTOR, which is tuberous sclerosis protein, TSC1 and 2, they are present in the lysosome, and mTOR remains inactive. Now, in the presence of amino acids, what happens is amino acids would activate a heterodimer complex proteins called as RAG GTPase. So the RAG GTPase recruits mTOR from the cytoplasm to the lysosome. Now, near the lysosome, there's another protein called MCRS1, which recruits a G protein called as Reb. Now, G protein Reb, when it combines or interacts with mTOR, it gets activated. But G protein Reb is kept in check by this tuberous sclerosis, TSC1 and 2 complex. So as long as TSC1 and 2 complex are in the lysosome, localized to the lysosomal membrane, Reb is kept in check and Reb does not interact with mTOR. Now, in a scenario like here, we can think of abundance of calories, right? The person's not restricting calories. So adequate amount or even abundant amount of amino acids and insulin is present. That leads to activation of mTOR. And how does it happen? The TSC1 and TSC2 proteins dissociate from Reb and move away from the lysosome. Insulin can also promote the detachment of this PRAS40 protein that is a part of mTOR complex. So PRAS40 gets dislocated or removed from mTOR. Now mTOR is able to interact with Reb, which is a G protein, Reb GTP and mTOR. So that activates mTOR. Now mTOR is active. We know what happens with an active mTOR. Active mTOR inhibits autophagy, but it promotes protein synthesis, anabolic reaction. So it causes protein synthesis to happen, whereas it's inhibitory to autophagy. Not only protein synthesis, mTOR has the capacity to activate or promote clonal expansion of pre-adipocytes. We have pre-adipocytes here. So pre-adipocytes clonally expand and then become adipocytes and mature adipocytes. So this activity is also promoted by mTOR activation. mTOR is able to activate certain transcription factors known as CEBPs. Now CEBP activation promotes clonal expansion and mature adipocyte formation. So mTOR appears to be necessary and sufficient for adipocyte differentiation. So that leads to fat accumulation and obesity. All those are associated with mTOR signaling pathway. Research shows that inhibition of mTOR actually promotes immune cell function in the elderly. And not only that, there are several other research in animal models and in human subjects that shows that inhibition of mTOR actually promotes health, prevents chronic diseases, and reduces markers of senescence. So mTOR inhibition, either by calorie restriction, exercise, or pharmacological interventions such as rapamycin or metformin can promote health and prevent the onset of chronic diseases and improve health span. So here again, there's a new study came out in 2019 that talked about how all these pathways, AMPK, mTOR, all converge and then point to one main pathway or one main process within the cell, which is autophagy. So mTOR inhibition promotes autophagy. mTOR activation promotes protein synthesis. So protein synthesis, anabolic mechanisms are activated when mTOR is active. When mTOR is inactive, that's when autophagy is promoted. So what is autophagy and how is it promoted, right? So once again, energy stress by calorie restriction or exercise, or even pharmacological agents which can mimic energy stress, such as rapamycin, mTOR, metformin, or spermidine, can all activate autophagy pathway. So activation of autophagy pathway has been linked to improved metabolic processes, improved cellular homeostasis, improved organelle quality control, all of which, as you know, can lead to anti-aging effects and delay the onset of chronic diseases. So autophagy is a conserved process. Again, we see autophagy happening in worm and all the way to humans. And the process primarily sequesters cytoplasmic constituents that are not needed or that are excess, and it breaks them down and recycles it, similar to cleaning our house, taking off unwanted stuff, recycling it, which improves the quality and nature. The same thing occurs within the cell. So autophagy, because it's a recycling process, can serve diverse purposes. So if you see, we have proteins, say those protein aggregates are not needed by the cells. They'll be broken down by autophagy pathway, and that will generate these small units, which is the amino acids. So those amino acids can then enter into the energy metabolism and form ATP, or can join the systemic pool of amino acids that are present, take part in protein synthesis. Same type of reactions happen when lipids undergo autophagy process. So complex lipids being in excess can cause oxidative stress within the cells. They can be taken, broken down, used for energy, because autophagy process breaks them down and then they can be recycled. Same happens with glucose, glycogen breakdown, glucose unit used for energy production. Not only macronutrients, compounds such as ferritin can be broken down, recycled, and used for other purposes. So this is a recycling process which not only clears the cell of the junk, but also promotes anabolism and use of these reconstituted or recycled material for other purposes. So with age, the autophagy process declines, and several of aging-associated diseases have been attributed to decline in autophagy. So I thought I'd just briefly explain the process of autophagy. So autophagy happens when an initiation membrane is generated within the cell. This happens in the cytoplasm. So the entire cell, imagine this is the cytoplasm where this autophagy process happens. Now, initiation leads to nucleation. The membrane called phagophore now starts to grow. I can find my, the membrane starts to grow. While the phagophore membrane is growing, we can see that, say there are mitochondria which are dysfunctional. or there are excess mitochondria which the cell wants to get rid of. So there are protein aggregates which are no longer needed. Sometimes many organelles, such as peroxisome, ribosome, all of these, they are excess or damaged in some way. They can be sequestered, pulled into this growing phagophore right here. Now, how are they pulled into the phagophore? We have adapter proteins such as P62 and NBR, which can tag on to these organelles that need to be, or the proteins that need to be destroyed. They just pull them, tag onto them, pull them into the growing phagophore. Now, all the unwanted materials, which in most cases, if they are in excess, they'll be generating reactive oxygen species, which we all know, superoxide, hydrogen peroxide, peroxynitrate, all those can be generated by excessive and dysfunctional organelles. So when they are removed, we clear the cell of reactive oxygen species. The reactive oxygen species is important signaling molecules themselves, but they have to be maintained in a balance. Excess of reactive oxygen species causes an oxidative stress, so they can be cleared by this process of autophagy. So once the organelles and unwanted material are sequestered into this autophagosome, the autophagosome then fuses with the lysosome. And lysosome, as you all know, has acid hydrolases that are present, which can chop down, break down these organelles and excess material. And then that will lead to nutrient recycling. Okay, I think I'm a little behind on my, yeah, so that results in nutrient recycling. So impairment or dysregulation of this autophagy process is reported in many cell types, in humans and animal models. And here is the cardiovascular aging. And you can see how the processes that we talked about, the three signaling pathways, mTOR, AMPK, insulin signaling, all these can, mTOR hyperactivation, reduced activity of AMPK, increased signaling through insulin receptor. And we didn't talk much about sirtuins. The sirtuins are another important signaling molecules, especially in the context of aging. And it involves mitochondrial signaling as well. And it's a vast topic. I didn't think that I could do justice to that topic here because it's a short lecture. So just letting you know that it's another important signaling pathway. All these pathways, with certain hyperactivation, for example, in case of mTOR or reduced activity of AMPK, can all result in impairment in autophagy. So this happens with age. With age, there's decline in autophagy that results in aberrant signaling as well as increased reactive oxygen species, which have all been identified as the trigger or signal for the onset of aging-associated diseases. And you can see some people have this mTOR-centric model, which shows that mTOR hyperactivation leads to inhibition of autophagy and hypertrophy and signal resistance, which can all point or trigger, push cells, cause them to become aberrant and promote various aging-associated diseases, especially cancers. And then there's also the ROS-centric model, reactive oxygen species, and they say that ROS generation, which is not cleared because of impaired autophagy, that leads to the onset of aging and aging-associated diseases. So as an overview, you would know by now that how these signaling, nutrient signaling pathways come together, and when they are dysregulated, it can promote age-associated diseases. That will, limit our health span. And if we are able to find out ways or identify ways through which we can inhibit these signals or activate them appropriately, that could lead to enhanced or improved health, delay the onset of chronic illnesses, and improve health span. So the signaling pathway that we saw is very, a tiny portion of massive signaling network that is present within the cell. Even what you're seeing now, this signaling pathway is not, it's not represent, or it doesn't represent all the signaling events that occur within our body. The portion that we saw was only nutrient signaling, which is shown here. Apart from that, we have hormonal signaling, oxidative stress-mediated signaling, signals that generate from or originate in the mitochondria, signal that we get from the nucleus. And even within this, not all protein, not all players are shown. But you could see how concerted and intricate and how highly regulated these mechanisms are. And if we identify the pathways that go awry with age and prevent them, we will be able to extend lifespan and health span. With that, I conclude. And thank you very much for your time and for listening. Here are my references. And I have a couple of books which you can refer to as well. Thank you.